This is the review for Unit 8, and I will be going through the learning objectives on page 107. This chapter looked at genetics, so how certain traits are inherited from one generation to the next. The first objective says, recognize dominant and recessive alleles and distinguish between genotypes and phenotypes. Before getting into this objective, let's first go over some important terminology. First off, remember that every single trait that you have, whether it's a physical disorder or some sort of physical trait, it's controlled by your genes. And genes are simply sections of your DNA that control for a trait. And every individual, this is really important, every individual has two copies of each gene. And that is because you got one copy from one parent and you got the other copy from the other parent. However, do you think that each gene that you got from each parent is exactly the same? Probably not. There's probably slight variations in those two genes. So these two genes would be found on these homologous chromosomes, as you can see in this picture here. However, the genes can vary slightly, and that variation is the result of slight differences in the DNA sequence. These variations of a gene are called alleles. And there's two main types of variations of genes that can occur. And one variation is called a dominant allele, as we represent with a capital letter. And if this dominant allele is present, if one of the two copies of the genes that you have is a dominant allele, then it will always be expressed. You will always show that dominant allele, whatever physical characteristic it would create. However, another type of allele that can be present is called a recessive allele. And we usually use a lowercase letter to designate a recessive allele. So this recessive allele is not going to be expressed if it's paired with a dominant allele. So if you inherit one dominant allele from one parent and a recessive allele from another parent, that recessive allele will never be expressed or shown in you because that dominant allele will overpower it. It will dominate it. Okay, so the only time that you will ever express or show a recessive allele is if you inherited both a recessive allele from your mother and from your father. Okay, so only time a recessive allele or the characteristic that it controls for is shown is if you have two recessive alleles. Both copies of the gene are the same. So we have two different types of alleles, dominant or recessive, and as I mentioned, each individual has two copies of a gene, so they have two alleles. So if we're looking at the allele combination an individual has, or in other words, their genetic makeup, we call this the genotype. So each individual will have two alleles that control for a trait. Both of those alleles could be dominant alleles, and we call that homozygous dominant. Homozygous, homo means the same. So both alleles are the same and they're dominant. Okay, so it's pretty easy to remember. However, it's possible that you inherited a dominant allele from one parent and a recessive allele from another parent, so then you would be heterozygous. And this means that you have a dominant allele and a recessive allele. Hetero means different, so the two alleles are different. If you have two recessive alleles for a trait, then you are homozygous recessive. Okay, and those are the only three genotypes that are possible. Another important term that you should know is true breeding. An individual is true breeding if they have two of the same alleles. So you could be true breeding for the dominant allele and be big A, or you could be true breeding for the recessive allele and be little a. We call this true breeding because an individual that is true breeding, for example, for the dominant allele, well, what can they give to their offspring? The only thing they could give their offspring is the dominant allele. So they're true breeding for that allele. Another important term you should know is carrier. Another word for carrier is just a heterozygote. So heterozygote and carrier mean the same thing. Heterozygotes are sometimes called carriers because they carry the recessive allele, but they do not express it. Many disorders are carried on the recessive allele, so if an individual is a carrier for that recessive allele, they will not express the disorder. They will not show that they have the disorder, and they might not know they have the disorder. but they are a carrier of it because they can still pass it on to their offspring. And if they pass that allele on to their offspring and their spouse also does, then their offspring could actually have the disorder. Another term that you should be familiar with is phenotype. So we could describe somebody's genotype, their genetic composition, or we could describe their phenotype, which is just what is their physical appearance? How do they look? And then finally, the term wild type. This is important. This is the most common phenotype in a population. We'll usually describe it as the wild type. Important thing to note here is that the wild type phenotype, it could be caused by a dominant allele or it could be caused by recessive allele. So it's possible that a recessive allele is more common in a population than a dominant allele. Okay, so now that you understand the terminology and you're able to recognize dominant and recessive alleles, it says that you need to be able to distinguish between genotype from phenotype. So when we're looking at a monohybrid cross, so this is a cross involving one trait, so you only have one gene with two possible alleles. For example, let's look at the trait plant color. Let's make sure we can tell the difference between genotype and phenotype. So let's say we have our dominant allele causes the plants to be green. Our recessive allele causes them to be white. Well, what are the possible genotypes that could exist in this plant population? Well, they could be homozygous dominant, they could be heterozygous, or they could be homozygous recessive. What type of phenotypes would be associated with each of these genotypes? Well, if they're homozygous dominant, they're going to express the big letter because that's all they have, so they'll be green. If they're heterozygous, they're going to again express the big letter because the big letter dominates the recessive letter, or the little one, and they'll also be green. If they're homozygous recessive, they will be white. An important thing to note here is that if an individual is expressing the recessive trait, then you automatically know their genotype because they have to be homozygous recessive. However, if I was holding a green plant in front of you, because that is the dominant trait or called by the dominant allele, you don't know if they're homozygous dominant or heterozygous. So you can't tell the genotype of an individual that is expressing the dominant phenotype. You can't tell what their genotype is just by looking at them. So that's an important thing to note there. So objective 2 says describe the phenotypic ratios among offspring in monohybrid crosses involving complete dominance. So in order to do that, you have to know how to do a Punnett square. And some simple rules for solving any type of Punnett square is to follow these steps. First, make a key for the dominant and recessive allele. What does each letter equal? Determine the genotype of the parent and figure out the possible gametes or sperm or egg they could produce. Next, construct a Punnett square with possible gamete types across the top and the left side, and then fill in the boxes with the possible genotypes of the offspring. All right, so you just always follow these steps. So let's do an example here. Here, we already have our key written out. Big G equals green, little g equals white. We already know what parents are being crossed here. Heterozygous is being crossed with another heterozygous. Well, let's now figure out what gametes each parent can make. We know that gametes are always haploid. They always have only one copy of a gene. So the gametes that this first parent could make would either have a big G in it or they would have a little g. So one or the other. A gamete can never have two of the same letter in them. The other parent could make also a gamete possible type of G, big G, or little g. So when you make your Punnett square, all you do is you write the possible gametes for one parent across the top, possible gametes for the other parent down the side. So we get big G, little g, big G, little g. Then you make a box, make a little row and column for each gamete, and then you just bring these together inside your box. And we would get big G, big G, big G, little g, big G, little g, and little g, little g. So we could express these results in a couple of different ways. One we could do is a phenotypic ratio. So how many green to white? Well, we would have 3 green, 1, 2, 3 to 1 white. Never give your answer as a ratio unless you're asked to. Normally, you're going to be asked to express this as a probability. So what's the probability that they'll be green or white? Well, the probability they'll be green is 3 out of 4, or 3/4, or 75%. Probabilities are always expressed as a fraction or a percent. What's the probability they'll be white? Well, we have 1 out of 4, so that'd be 1/4 or 25%. You could also do the genotype probability. There'll be 25% chance, or 1/4, that they'll be homozygous dominant. 2 out of 4 would be heterozygous, so that's 50%, and only 1/4 or 25% would be homozygous recessive. All right, so I think we're pretty good with monohybrid crosses. So let's move down to objective 4, where it says construct Punnett squares and predict phenotypic ratios in offspring from dihybrid crosses. So a dihybrid cross is no different than a monohybrid cross, except you're looking at 2 traits. So you might be looking at, for example, eye color versus wing type and wing type. So you're looking at how these 2 traits are inherited together. And one important thing to note, though, is that eye color does not affect the inheritance of wing type. So these 2 traits are inherited independently of each other. So again, you want to come up with a key. So we'll use big E equals red eyes, little e equals sepia eyes, big W equals normal wings, little w equals vestigial wings. They're these little tiny wings here. All right, let's say we cross these 2 parents. So this parent is heterozygous for eye color and heterozygous for wings, and the same with this parent. What are the possible gametes that can be produced? So this usually gets really confusing for students, but don't make it more complex than it has to be, right? So we know from before that each gamete gets 1 copy of each gene. So all you got to do is figure out all the possible combinations of the 2 different letters possible. So they're going to get 1 copy of the E gene and 1 copy of the W gene at the same time. So to figure out all the possible combinations, you can just look at all the possible pairings, right? So I take the first letter of my first pair and just pair it up with both letters of the second pair. So big E with big W, big E with little w. And then I do the same thing with the second letter of this first pair. Little e with big W, little e with little w, and these are all my possible combinations of E's and W's. You have to do that to figure out the possible gametes. If I want to figure out the possible gametes of the other parent, it's going to be the same thing. Then to do the Punnett square, you do the same thing as before. You put the possible gametes of one parent along the top, the possible gametes of the other parent along the bottom here. And then you create your box for each one. And then you can make all your possible offspring in the inside here. So you bring these 2 gametes together. If these 2 fertilize each other, what would we get? Big E, big W, big W. Bring the next ones together, next ones, and just keep going. Okay, so now we have those all done. Now we can look at the phenotypic ratio, or you could figure out the probabilities as well. So if we looked at the phenotypic ratio, we just add up how many boxes we have. What are red eye and normal wings? Well, red eyes are big E, normal wings are big W, so we're just looking for 1 big E and 1 big W at least, and that's all of these. So there's 9 of them. If we're looking at red eye and vestigial wings, red eye again is big E. To be vestigial, they need to have 2 little W's. So of what's left, what do we see? It's these ones here. And then for CPI and normal wing, if we look for that, CPI, they need to have 2 little e's. To have normal wings, they have to have a big W. So we can see that here. And then we only have 1 left down here, and that one is sepia and vestigial wings. So that's 1. So we'd have a ratio of 9 to 3 to 3 to 1. If you wanted to figure out the probabilities, all you would do, so like what's the probability there'll be red eye and normal wings? Well, there's 9 of them, and it's 9 out of 16, so your probability would be 9/16. Let's do another practice of dihybrid crosses. So let's just say we have red petals and pink petals. So petal color is 1 trait. And then we're looking at leaf color as well. So we have green leaves and purple leaves. Big G and little g. So remember, our parents are diploid, so they're going to have 2 copies of each gene. And let's say one parent is true breeding for red petals and true breeding for purple leaves. So they big R, little g, little g. The other parent is heterozygous for both traits. Well, let's look at the possible offspring they could make. Again, you just figure out the possible gametes. If we do our method here, we would get the same gametes created each time. So this parent can only create one possible type of gamete, big R, little g. The other parent, we can pair the big R with the big G, big R with the little R with the big G, and little r with the little g. So we have 4 possible gametes. So whenever we do our Punnett square, we want to write the possible gametes along the top and the side. Since this parent can only make one type, just write it once. That saves you a lot of time. The other parent can do it 4 times, make 4 different ones, so we write all those out. Put a box for each one of these gametes. Bring them together to get the offspring. So we brought these 2 together. We get big R, big G, little g. Bring these ones together, big R, little g, so forth. And we get that. Well, what if we want to know what's the probability the offspring will have red petals and purple leaves? For the offspring to have red petals, they have to have at least 1 big R. To have purple leaves, they have to have, since it's recessive, they have to have 2 little g's. Well, do you see any here with 2 little g's and at least 1 big R? Well, I see one there and there. So what's our probability? It's 2/4, or in other words, 1/2 or 50%. And that is your answer. All right, the last objective says interpret pedigrees and determine specific genotypes through pedigree analysis. Pedigrees are kind of cool little things because they show the inheritance pattern of certain traits in a family. And the whole goal of using a family pedigree is to determine if the trait that's shaded in is caused by a dominant or recessive allele. So is it caused by a big letter or is it caused by a little letter? Or in other words, is it caused by a big letter or is it caused by having 2 little letters, right? So in order to do these pedigrees, first you need to understand the symbols here. Males are always squares, females are always circles. A mating pair is shown by a line between the individuals. You can see there's 3 mating pairs in this family. You can see a line coming down from the mating pair means that they have children, and then you can see that these parents have 3 children. These parents have 2 children. And then individuals that are affected, or in other words, have the trait we're interested in, are colored in, and those that are not affected or do not have the trait we're interested in are not shaded. So an important thing to remember, the shaded shapes indicate the trait that we're interested in. It does not tell us whether it's dominant or recessive. That's something that we have to figure out on our own. So some important tips for solving family pedigrees. First thing, in every pedigree that you will see, if it's one that has enough information to tell whether the trait's due to a dominant or recessive allele, you're going to find 2 parents that are alike. So either both parents are shaded or both parents are not shaded, and they have at least 1 child that's different from them. You want to find that family on every pedigree because that will help you figure out with certainty if the trait is due to a dominant or recessive allele. And that is because the only way that you could see this pattern on the left here or this pattern on the right is for both parents to be heterozygous. Both parents must be heterozygous to create an offspring that looks different than both of them. Okay, so that's an important thing to remember. Another important tip for solving family pedigrees, if both parents are unaffected, as you see over here, but they have a child that has the trait, that means that the trait, or in other words, a shaded individual, has to have 2 recessive alleles. So the trait has to be caused by the recessive allele. If both parents are affected, but their child is unaffected, as you see here, then that means the trait that we're interested in, the colored in individuals, must have the dominant allele. The trait is due to a dominant allele. Individuals then, in that case, that are not shaded in, must have 2 recessive alleles. So on the left here, we know these parents have to be heterozygous. Their offspring is homozygous recessive. They got a little a from each parent. These individuals, since they look like the parents, they have to have a dominant letter. But they could have gotten a big A, So a big A from each parent, or they could have gotten a big A from one parent and a little a from the other parent. And in both cases, they would be expressing the dominant trait. So there's not enough information to tell us whether these individuals are homozygous dominant or heterozygous. And the right side here, both these parents have to be heterozygous. So since they're heterozygous, they have the dominant allele. So whatever they look like, so here they're shaded in, that means shaded in is caused by the dominant allele. We know that any individual that looks different than those parents, like these offspring here, must be homozygous recessive. This child here looks like the parents, so it also must have a big letter. And in this case, again, we don't have enough information to know what the other letter is. All right, so let's look at an example. Here we're looking at the inheritance of methemoglobinemia. And we want to know whether this is due to a dominant or recessive allele. So first, find the parents that look alike that have one child that's different than them, at least one child that's different than them. And here we can see that. Both parents have to be heterozygous for this to happen. So now we know that big H has to equal unshaded. And little h equals shaded. Because we know that we know these parents are heterozygous, and heterozygotes always express the dominant allele. So this means that big H equals not being shaded in. We know every individual that's shaded in has a trait we're interested in, which is methemoglobinemia. So is methemoglobinemia due to a dominant or recessive allele? Well, as we can see here, it's due to a recessive allele. So our answer there would be recessive. And we know that every individual that has this condition must have 2 recessive alleles. So we already could fill out half of our pedigree just by knowing that. Then it asks us, what is the genotype of the female marked by the arrow? So this female here. Well, we know that she's not shaded in, so she must have a big H. We know that one of her letters is a big H, but what's the other letter? Well, let's just look up at her parents here. What is the genotype of this parent here? Well, we also know that this parent must have a big H, right? And we know that they created a child with 2 little h's. So this parent must also have a little h, so they must be heterozygous. If we look at this female too, she has a big H that she could have gotten from this parent. But what did she have to get from her father here? The only thing she could have got from him is a little h, so she must be heterozygous. We know that with certainty, that she's a heterozygous. What about this individual here? This is our last individual left. Let's just do this for fun. Well, this individual has to have a big H, right? Because they're not shaded in. But they could be big H, or big H, little h. So there's not enough information. They could be homozygous dominant, or they could be heterozygous. Here, we're looking at the inheritance of spherocytosis. Again, look for the parents that are similar and have an offspring, at least one offspring that's different than them here. Is spherocytosis due to a dominant or recessive allele? Well, what do we know about those parents? How could they create a child that's different than them? They have to be heterozygous. And again, if you want to, I think this is always the easiest way to just do this, is to make a little key. So we know now that these parents are expressed, since they're heterozygous, they have to be expressing the big letter, and that equals shaded. And the little letter would have to equal unshaded. So what does that mean? Well, every individual with a big A has spherocytosis because they're shaded in. So is spherocytosis due to a dominant or recessive allele? Well, it has to be due to a dominant allele. Another way that we know this is we know if 2 parents have the trait, but their child doesn't, then the trait's due to a dominant allele. What is the genotype of the male marked by the arrow? So this male we need to figure out. Well, we know he's unshaded, so unshaded individuals are expressing the recessive allele, so he must have two little a's. So he's homozygous recessive or little a. If the parent in row 1, the parents in this row had another child, what's the probability they would have spherocytosis? So we know this parent's homozygous recessive because they're unshaded. And we know that having the trait is caused by the dominant allele. So not having it has to be caused by the recessive allele. They're opposites. This individual, this mother, has spherocytosis, so we know she has to have at least one big A. And we know that her other letter has to be what? Could it be big A, big A? Well, if we look at her offspring, we can see that one offspring here has two little a's, so she must also at least have one little a, so she is heterozygous. So what's the probability their child would have spherocytosis? You just do a simple monohybrid cross, and we can see that half, 2 out of 4 of their children would have spherocytosis, so the probability is half or 50%. Hopefully these family pedigrees are now easier for you to solve, and good luck studying for your exam.